In the quiet moments of our lives when we seek solace and stillness, we often turn to the natural world. We walk through a forest, we sit in a garden, we rest under the shade of a great tree, and in these moments we are surrounded by a silent and ancient kingdom, a kingdom that is so ubiquitous, so fundamental to the fabric of our world that we often cease to see it at all. This is the kingdom of plants. They are the green, breathing backdrop to our hurried human lives, the silent scenery against which the drama of the animal world unfolds. We see them as passive, as decorative, as objects, but we rarely see them for what they truly are, a vast, complex and deeply ancient form of life, a civilization of beings that experience the world and act upon it in ways that are profoundly different from our own. The story of plants is the story of our world. It is a story that begins in the deep primordial oceans over 3 billion years ago, with a single microscopic cell that performed a quiet chemical miracle that would forever change the destiny of our planet. It is a story of a great and perilous colonization, the slow, courageous journey from the water to the barren, rocky land. It is a story of incredible innovation, of the invention of the root to mine the earth, of the leaf to drink the sun, and of wood to conquer the sky. It is a story of partnership and communication, of a delicate, ancient dance with the animal world, and of a hidden subterranean network that connects an entire forest in a silent, secret conversation. Tonight, we will embark on a journey into this silent kingdom. We will slow ourselves down, quiet our minds, and learn to see the world on a different timescale, the timescale of a plant. We will witness the slow, graceful, and deliberate movements of a vine as it seeks the sun. We will decode the complex chemical language of a flower's scent, and we will learn that plants are not passive objects, but active and sensitive beings. They can sense the quality and direction of light, they can feel the touch of an insect's feet, and they can communicate with each other in sophisticated and surprising ways. This is a journey that will challenge our most basic assumptions about what it means to be alive, what it means to sense, to communicate, and to be intelligent. It is a journey that will reveal that the green world around us is not a static backdrop, but a dynamic, active, and deeply interconnected web of life. It is a story that will, in the end, bring us back to ourselves, revealing our own profound and total dependence on this quiet, photosynthetic kingdom. For plants are the lungs of our planet, the foundation of our food, and the source of our medicines. To understand the secret life of plants is to understand the secret of life itself. Our story does not begin in a forest or a garden. It begins in a world that would be utterly alien and unrecognizable to us. A world from the deep, distant past over 3 billion years ago. Let us quiet our minds and travel back to this primordial earth. The sky is not blue, but a hazy, pinkish orange. The continents are not the familiar shapes we know, but a single, vast and barren supercontinent of dark volcanic rock. There are no trees, no grasses, no flowers. There is no soil. The land is a sterile and silent desert. The oceans are not blue, but a murky olive green, rich in dissolved iron. And the air, the very atmosphere of this ancient world, is a toxic and suffocating mixture of nitrogen, carbon dioxide, methane, and sulfurous gases. There is almost no free oxygen. For a human, this world would be instantly fatal. For over 1 billion years, life on this planet had been a purely microbial affair, a simple and singular drama playing out in the shallow, sunlit waters of the oceans. The only inhabitants were tiny, single-celled organisms, prokaryotes, the ancestors of modern bacteria. They were simple, anaerobic beings, living a life without oxygen, deriving their energy from the chemical reactions of the planet's primordial soup. They were the first tentative expressions of life's potential, but they were living in a world of scarcity. Their existence was limited by the availability of these chemical energy sources. Life had reached a bottleneck, an energy crisis. For life to take the next great leap, for it to become the complex, diverse, and abundant force that we know today, it needed to find a new and virtually limitless source of energy. That source of energy was shining down on the planet every single day, a vast, untapped nuclear furnace in the sky, the sun. The challenge was to find a way to capture its energy, to convert the fleeting ethereal energy of a photon of light into the stable, chemical energy that a living cell could use. The story of photosynthesis is the story of how life solved this problem. It is the story of a single, microscopic cell that performed a quiet chemical miracle, a miracle that would not only solve the energy crisis, but would also completely and irrevocably transform the entire planet. Our protagonist in this story is a humble, single-celled organism called a cyanobacterium. We can imagine it, floating in the warm, shallow waters of a primordial sea. It is a simple creature, but inside its tiny cellular body, a revolutionary experiment is taking place. It is beginning to evolve a new and remarkable kind of molecule, a pigment. This pigment has a special and almost magical property. It can absorb the energy of a photon of light. This molecule is chlorophyll. The evolution of chlorophyll was the first step. But absorbing the energy of light is not enough. That energy must be captured and converted into a stable, usable form. The cyanobacterium evolved an intricate and beautiful molecular machine to do just this. This machine, which we now call a photosystem, is a complex of proteins and chlorophyll molecules, all embedded in the cell's membrane. When a photon of sunlight strikes a chlorophyll molecule, its energy is captured and funneled with incredible speed and efficiency to a special reaction center at the heart of the photosystem. Here, the captured solar energy is used to perform a truly audacious act of chemical alchemy. The cyanobacterium needed a source of electrons to power its metabolic processes. The other, more primitive photosynthetic bacteria of the time were using easily accessible, but scarce, electron donors like hydrogen sulfide. But the cyanobacterium learned to do something far more difficult and far more revolutionary. It learned how to take the most abundant and stable molecule in its environment, water, H2O, and to split it apart. This is not an easy thing to do. The water molecule is incredibly stable. It does not want to be split. It requires a huge input of energy, but the cyanobacterium, using the power of the sunlight it had captured, evolved a special enzyme, a water-splitting complex that could wrench the electrons away from the water molecule. This act of splitting water was the great innovation, the killer app of photosynthesis. It gave the cyanobacteria access to a virtually infinite and ubiquitous source of electrons, freeing them from the scarcity that had limited all other life. They could now live almost anywhere there was sunlight and water. But this revolutionary act had a byproduct. When you split a water molecule, you are left with protons, electrons, and a waste product. That waste product is oxygen. For the cyanobacterium, oxygen was a toxic, corrosive poison. It was a dangerous chemical that reacted with and damaged the delicate machinery of the cell. The cyanobacteria had to evolve new protective enzymes to defend themselves against their own toxic waste. For millions of years, this new, oxygen-producing photosynthesis was a local affair. The oxygen produced by the cyanobacteria did not immediately build up in the atmosphere. Instead, it reacted with the vast amounts of dissolved iron in the oceans. The oxygen combined with the iron to form iron oxide, rust. This rust then precipitated out of the water and settled on the ocean floor, forming vast, thick layers of rock that we can still see today as the banded iron formations. For hundreds of millions of years, the oceans literally rusted. The cyanobacteria were slowly, patiently terraforming their own planet, their toxic waste product being safely locked away in the rock. But eventually, after about 1 billion years of this continuous planetary-scale oxygen production, the dissolved iron in the oceans was all used up. There was nowhere else for the oxygen to go. And so, it began to bubble out of the oceans and into the atmosphere. This was the beginning of what is now called the Great Oxidation event, or more dramatically, the oxygen catastrophe. For the other anaerobic life forms that dominated the planet, this was an apocalypse. Oxygen was a deadly poison to them. As the oxygen levels in the atmosphere began to rise, it caused a mass extinction, perhaps the greatest mass extinction in the history of the planet. The old anaerobic world was dying, poisoned by the waste product of a new and powerful form of life. The cyanobacteria, in their success, had inadvertently destroyed the world they were born into, but life is resilient. In the face of this great crisis, a new kind of life evolved, a life that could not only tolerate oxygen, but could learn to use it. A new and far more efficient form of metabolism, aerobic respiration, evolved. This new metabolism used oxygen to burn the sugars produced by photosynthesis, releasing a huge amount of energy. This paved the way for the evolution of a new, more complex kind of cell, the eukaryotic cell, the cell that would eventually give rise to all plants, all animals, and all fungi. To contemplate this first green spark is to meditate on the profound and planet-altering power of a single biological innovation. It is a story that reminds us that the world we inhabit today, the blue sky, the oxygen-rich air, the very possibility of our own complex air-breathing existence, is a direct consequence of this quiet chemical miracle that was first performed by a humble single-celled organism in a primordial sea. The cyanobacteria were the first and greatest engineers in the history of life. They invented a way to eat the sun, and in doing so, they created the world. Every plant we see today, from the smallest blade of grass to the tallest redwood tree, is a direct descendant of this ancient lineage. Every leaf is a tiny biological machine that is still performing that same sacred and world-changing chemistry. The story of photosynthesis is the story of the birth of our world. It is the quiet, green fire that powers almost all of life on Earth. For over 3 billion years, the entirety of life's drama had been an aquatic affair. The oceans, the lakes, and the shallow seas teemed with a rich and diverse microbial life, and later, with the first simple, multicelled algae. The water was a gentle and forgiving cradle. It provided structural support, preventing the delicate bodies of these early organisms from collapsing under their own weight. It offered a stable and predictable environment, buffering them from the wild temperature swings of the atmosphere. It was a constant, nourishing bath, providing all the water and dissolved nutrients a cell could need directly through its cell wall. And it protected them from the most dangerous threat of all, the harsh, unfiltered ultraviolet radiation from the sun, which would have shattered the delicate molecules of life. The oceans were a comfortable, self-contained, and complete world. The land, by contrast, was a realm of death. Let us for a moment contemplate the land of the early Paleozoic era, around 500 million years ago. It is a world of stark and brutal simplicity. There is no soil, only bare rock, gravel and sand, the raw pulverized crust of the earth. The sun beats down from a sky that is a deeper, harsher blue than the one we know, its light unfiltered by a fully formed ozone layer. The air is thin and dry. The days are scorchingly hot, and the nights are freezing cold. And there is no water save for the rain that falls and quickly runs off the bare rock, or the moisture that clings to the edges of the seas and the freshwater lakes. For an aquatic organism, the land was a desert, a place of desiccation, of crushing gravity, of extreme temperatures and of deadly radiation. It was an alien planet, a hostile and seemingly uninhabitable frontier. And yet, it was also a world of immense opportunity. The land was a vast empty continent, a blank slate waiting to be written upon. The sunlight that fell on the land was a powerful and untapped source of energy. The atmosphere was rich in carbon dioxide, the essential raw material for photosynthesis, and there were no herbivores, no creatures that had yet evolved to eat the first pioneering plants. The land was a promised land, a world of unlimited space and resources, if only the immense challenges of survival could be overcome. The story of the colonization of the land is the story of a great and perilous journey, the story of the quiet courage and the remarkable innovations of the first, humble plants that dared to leave the safety of the water and venture out onto this hostile shore. The pioneers of this great migration were likely the descendants of the green algae, simple, multicelled organisms that lived in the shallow freshwater pools at the edge of the land. We can imagine these pools drying up periodically in the heat of the sun, exposing the algae to the air for brief, stressful periods. This was the training ground. In these fluctuating environments, natural selection would have favoured any alga that had a slightly better ability to survive these periods of exposure. Perhaps it evolved a slightly thicker outer layer, a primitive cuticle, to reduce water loss. Perhaps it evolved a greater tolerance for the damaging effects of UV radiation. Over millions of years, these small, incremental adaptations accumulated, preparing these aquatic plants for a more permanent life on the land. The first true land plants were likely small, simple, and unassuming organisms, something akin to modern-day liverworts or mosses. They did not have true roots, or stems, or leaves. They were just a simple, flat branching structure of green tissue, a thallus that clung to the damp rocks at the water's edge. Their lives were still intimately tied to the water. They had no internal plumbing, no vascular system to transport water from one part of their body to another. They had to absorb water directly through their surfaces like a sponge, and most importantly, their method of reproduction still depended on water. Their male reproductive cells, the sperm, were swimming cells, equipped with tiny whip-like flagella. They needed a film of water, a raindrop or a layer of dew to be able to swim to and fertilize the female egg. This is why, even today, we find mosses and liverworts primarily in damp, shady places. They are the living reminders of this first tentative step onto the land, their lives still tethered to the aquatic world of their ancestors. Let us contemplate the immense challenges these first pioneers faced. The first and greatest challenge was water. On land, water was scarce and fleeting. The plant had to evolve a way to hold on to the water it had, and a way to find more. The evolution of a waxy cuticle was a crucial first step. This was a thin, waterproof layer that coated the surface of the plant, like a layer of wax on a car, dramatically reducing the evaporation of water from its cells into the dry air. But this solution created a new problem. A waterproof coating also prevents the plant from taking in carbon dioxide from the atmosphere, the essential gas it needs for photosynthesis. The solution was another brilliant innovation, the evolution of tiny adjustable pores in the cuticle called stomata. Each stoma is a microscopic mouth surrounded by two guard cells that can open to allow carbon dioxide to enter and close to prevent water from escaping. The plant could now breathe without dying of thirst. The second great challenge was gravity. In the water, the plant was buoyed up, its weight supported by the dense medium around it. On land, in the thin air, the plant had to support its own weight. The first, small, ground-hugging plants solved this problem by simply not getting very big. But for plants to grow taller, to compete for sunlight, they needed to evolve a form of structural support. This led to the evolution of specialized rigid cells, and eventually to the invention of lignin, the complex polymer that makes wood hard. Lignin was the key to building the first true stems, the biological scaffolding that allowed plants to grow upright and to defy the constant crushing pull of gravity. The third challenge was reproduction. The reliance on swimming sperm was a major limitation, tying the plants to the water's edge. The great evolutionary leap that truly freed plants from their dependence on water was the invention of pollen and the seed. Pollen was a revolutionary new way of transporting the male reproductive cells. Instead of a fragile swimming sperm, the male genetic material was packaged inside a tiny, tough, and desiccation-resistant grain that could be carried for vast distances by the wind. The plant no longer needed water to reproduce, it could use the air itself. The seed was an equally profound innovation. Instead of a single, vulnerable egg, the fertilized embryo was now packaged inside a protective, nutrient-rich container, a tiny, portable life support system. The seed could protect the dormant embryo from harsh conditions, and it could wait, sometimes for years, for the perfect moment to germinate. The evolution of pollen and the seed was what allowed the plants to finally conquer the dry interiors of the continents, to move away from the coasts and the rivers, and to colonize the entire planet. The colonization of the land by plants was one of the most significant events in the history of life on Earth. It was an act of planetary-scale engineering that fundamentally transformed the world. As the first plants grew and died, their decaying organic matter began to mix with the weathered rock, creating for the first time a rich, living layer of soil. The roots of these plants held the soil in place, preventing erosion. The vast new forest began to change the climate, increasing humidity and stabilizing temperatures. And most importantly, through the relentless quiet work of photosynthesis, they continued the work that the cyanobacteria had started, pumping vast quantities of oxygen into the atmosphere and drawing down huge amounts of carbon dioxide, cooling the planet and creating the stable, oxygen-rich air that all complex animal life, including ourselves, depends on. To contemplate this great colonization is to be filled with a sense of awe at the sheer, dogged tenacity of life. It is a story of a slow, patient and courageous journey from a world of comfort and safety into a world of immense hardship and immense opportunity. The first humble plants that clung to the damp rocks at the edge of the ancient seas were the true pioneers of our world. They were the architects of the green planet. Every forest, every meadow, every garden we see today is a testament to their ancient, perilous, and ultimately triumphant journey. As the first pioneering plants made their tentative journey onto the land, they faced a world of profound and challenging dualities. The resources they needed for life were now separated into two different and often distant realms. Above the ground in the airy sunlit world was the energy and the carbon they needed for photosynthesis. But below the ground, in the dark, hidden world of the rock and the newly forming soil, was the water and the essential mineral nutrients that were just as vital for their survival. The plant had to find a way to live in both of these worlds at once, to bridge the gap between the sky and the earth. The evolutionary answer to this challenge was a masterpiece of biological engineering, a completely new and revolutionary kind of organ. This was the invention of the root. The first land plants, the mosses and the liverworts, did not have true roots. They had simple, hair-like structures called rhizoids, whose only real function was to anchor the plant to the surface. They could not absorb any significant amount of water or nutrients. The plant was still, in essence, a sponge, absorbing everything it needed through its surface. But as plants grew larger and taller, as they evolved stems and leaves to compete for sunlight, this simple strategy was no longer sufficient. They needed a dedicated, specialized organ for anchorage and for absorption. They needed to grow down into the earth, just as they were growing up into the sky. Let us contemplate the quiet, hidden, and incredibly active life of a root. We are accustomed to thinking of the root system as a static, passive thing, a simple set of straws that suck up water from the soil, but this is a profound misunderstanding. The root system is a dynamic, sensitive, and intelligent organ. It is the plant's anchor, its mouth, and, in a very real sense, its brain. It is a complex, branching, and exploratory structure, constantly growing, constantly sensing, and constantly making decisions as it navigates the dark, complex, and heterogeneous world of the soil. The journey of a root begins with a single, downward growing tip. This tip is a place of incredible biological activity. It is protected by a small, tough, thimble-like structure called the root cap. The cells of the root cap are like the pioneers of a wagon train. They are constantly being sloughed off as the root pushes its way through the abrasive soil particles, and they are constantly being replaced from behind. The root cap also secretes a slimy substance called mucilage, which lubricates the path of the root, easing its passage through the soil. But the root cap does something even more remarkable. It is the site of the plant's sense of gravity. It contains specialized cells that hold tiny, dense starch grains called statoliths. These grains settle to the bottom of the cells, and this tells the plant which way is down, ensuring that the root always grows in the right direction, towards the promise of water. Just behind the root cap is the region of cell division, the meristem, where new cells are constantly being born. And just behind that is the region of elongation, where these new cells expand, pushing the root tip ever deeper into the soil. And then, just behind that, is the region where the root performs its most vital function, the region of absorption. Here, the surface of the root is covered in a fuzzy white carpet of millions of tiny, delicate, single-celled extensions called root hairs. Each root hair is a microscopic probe, dramatically increasing the surface area of the root system, allowing it to come into intimate contact with a vast volume of soil. It is through the delicate membranes of these root hairs that the plant drinks. The process of water absorption is a marvel of passive physics. The concentration of solutes inside the root cells is higher than the concentration of solutes in the soil water, and so, through the simple, inexorable process of osmosis, water flows from the area of high concentration, the soil, to the area of low concentration, the root, without the plant having to expend any energy at all. The absorption of mineral nutrients, however, is an active and highly selective process. The root is not just a passive straw, it is a discerning mouth. The cell membranes of the root hairs are studded with specialized protein channels and pumps, each one designed to recognize and transport a specific mineral ion, nitrate, phosphate, potassium, calcium, from the soil into the root. The plant can control which of these channels are open and which are closed, allowing it to take up the specific nutrients it needs while excluding those that are toxic or not required. But the root is more than just a mouth. It is a brain. It is constantly sensing its environment and making complex decisions. The root system is not a random chaotic tangle. It is a highly efficient foraging structure. A root will grow towards a patch of nutrient-rich soil and will proliferate there, sending out a dense network of fine branches to exploit the resource. If it encounters a patch of dry or nutrient-poor soil, it will grow straight through it, conserving its energy for more promising territory. If it encounters an obstacle, like a rock, it will grow around it. If it encounters the root of a competing plant, it may try to outcompete it, or it may grow away from it. The root system is constantly mapping its environment, creating a three-dimensional internal representation of the world beneath the ground. How does it do this? The root tip is an incredibly sensitive chemical sensor. It can taste the soil, it can detect the concentration gradients of dozens of different chemical compounds, of water, of nitrates, of phosphates, of toxins, of the chemical signals released by other roots and by soil microbes, and it can integrate all of this complex chemical information to make a simple, binary decision: grow or turn. This process, this constant slow-motion exploration and decision-making, is a form of intelligence. It is not the fast, centralized, neuron-based intelligence of an animal, but it is a slow, decentralized and distributed intelligence, a form of swarm intelligence that emerges from the collective behavior of thousands of individual sensing root tips. The world of the root is also a world of complex social interactions. The roots of a single plant are in constant communication with each other, but they are also in communication with the roots of other plants. Some plants engage in fierce, underground warfare, releasing toxic chemicals, a process called allelopathy, to inhibit the growth of their neighbours and to claim territory for themselves. But cooperation is just as common as competition. The roots of many plants can recognise the roots of their own kin, their close relatives, and will become less competitive, sharing resources more freely with them than they would with a stranger. And the social life of the root extends beyond the plant kingdom. The area of soil immediately surrounding the root, the rhizosphere, is one of the most densely populated and biologically active ecosystems on the planet. The root is constantly secreting a rich cocktail of sugars, amino acids, and other organic compounds into the soil. This is not a waste product. It is a deliberate act of cultivation. The plant is feeding and farming a specific community of beneficial bacteria and fungi. In return, these microbes help the plant to absorb nutrients, they protect it from pathogens, and they help to create a healthy soil structure. To contemplate the invention of the root is to be filled with a new appreciation for the hidden half of the plant world. It is to see the world beneath our feet not as a dead, inert substrate, but as a dark, rich, and complex universe. A universe that is being constantly explored, mapped, and shaped by the patient, intelligent, and life-giving work of the roots. The root is the organ that forever anchored the plant kingdom to the terrestrial world. It is the physical and metabolic bridge between the geological and the biological, the place where the dead minerals of the earth are drawn up and transformed into the living tissue of the green world. It is the silent, unseen, and essential foundation upon which the entire visible edifice of the terrestrial ecosystem is built. For millions of years after the Great Colonization of the land, the world was a flat green carpet. The first land plants, the mosses and their kin, were small ground-hugging organisms, their lives dictated by the need to stay close to the moisture of the earth. They had solved the problems of desiccation and reproduction, but they were living in a two-dimensional world. The third dimension, the vertical dimension, the space between the ground and the sky, remained an empty and unconquered frontier. The story of the tree is the story of the conquest of this frontier. It is the story of a revolutionary new material, a biological invention that allowed plants to defy the fundamental force of gravity, to build towering structures that could reach for the heavens, and to engage in a new and epic competition for the most precious resource of all, sunlight. This is the story of wood. Let us travel forward in time to the Devonian period, around 385 million years ago. The world is a warm, humid and green place, but it is a world without a single tree. The landscape is dominated by primitive fern-like plants and giant club mosses. In this world, a new and intense evolutionary pressure is beginning to build. As the land becomes more crowded, the competition for sunlight becomes fierce. A plant that could grow just a little bit taller than its neighbours would have a huge advantage. It could capture the unfiltered sunlight while casting its neighbours into shade, stunting their growth and securing its own dominance. The race for the sky had begun, but to grow tall, a plant had to solve the problem of gravity. The simple, soft tissues of the early land plants were not strong enough to support any significant height. They would simply collapse under their own weight. The solution was a remarkable new polymer, a complex, rigid, and incredibly strong substance called lignin. Lignin is one of the most abundant and important organic polymers on Earth, second only to cellulose. It is a complex, cross-linked molecule that infiltrates the cell walls of certain plant tissues, particularly the water-conducting cells of the xylem, and turns them into something completely new. It transforms a soft, flexible, living cell into a hard, rigid, and dead one. It turns plant tissue into wood. The evolution of lignin was a biological masterstroke. It provided, for the first time, a way to build a strong internal skeleton. Lignified xylem cells linked together could form a rigid, hollow tube like a steel pipe that was both incredibly strong in compression and efficient at transporting water. By bundling these tubes together, the plant could create a stem, a trunk, a biological skyscraper capable of supporting its own weight and reaching ever greater heights. The first trees were born. These first forests of the Devonian and the following Carboniferous period were strange and alien places. They were not filled with the familiar broadleaf trees of today, but with giant tree-sized versions of plants that we now know only as small, humble organisms. There were towering 100-foot-tall club mosses, like lepidodendron, with strange diamond-patterned bark. There were giant tree-like horsetails, calamites, with their segmented bamboo-like trunks, and there were the first primitive seed ferns, which were among the first plants to combine the strength of wood with the reproductive freedom of the seed. Let us contemplate the life of a tree. It is a life lived on a timescale that is almost unimaginable to our own fleeting human consciousness. A tree is a living monument to patience. It is a slow, deliberate, and multi-generational project of construction. It begins its life as a tiny, vulnerable seedling, a small speck of green on the forest floor. For years, or even decades, it may remain in this suppressed state, living in the deep shade of its parents, waiting for a gap to open in the canopy above. It is in a state of suspended animation, biding its time. Then, one day, an old tree falls in a storm or a branch breaks, and a small shaft of light penetrates the darkness of the forest floor. This is the moment the young tree has been waiting for. The light is a signal, a call to action. The seedling begins to pour all of its energy into upward growth. It builds its trunk, layer by layer, year by year. Each year it adds a new ring of wood, a new layer of xylem tissue. In the spring, when water is plentiful and growth is fast, it produces large, thin-walled cells, which form a light-coloured ring. In the summer, as growth slows, it produces smaller, thicker-walled cells, which form a dark ring. This annual rhythm of light and dark rings becomes a living, physical record of the tree's own life, a silent wooden diary that records the passage of the years, the good seasons and the bad, the droughts and the fires. The structure of a tree is a masterpiece, a biological engineering, a perfect solution to the problem of being a tall photosynthetic organism. The trunk is a marvel of structural efficiency, providing the maximum amount of strength with the minimum amount of material. The branching pattern is not random. It is a fractal, a mathematical pattern that repeats itself at different scales, a design that is perfectly optimized to arrange the leaves in a way that maximizes their exposure to the sun while minimizing the amount they shade each other. The leaves themselves are the solar panels, the food factories. The roots are the anchor and the mouth, gathering the water and nutrients from the soil. And the wood, the trunk and the branches, is the essential infrastructure, the skeleton and the plumbing that connects the leaves in the sky with the roots in the ground. But the wood is more than just a physical support. It is also a strategic reserve. The living tissues of a tree are actually just a very thin layer just beneath the bark. The cambium, where new cells are made, the phloem, which transports the sugars from the leaves, and the sapwood, the outer, active layers of xylem that transport water. The vast majority of the wood in a large tree, the heartwood, is actually dead. But it is not useless. The heartwood is often infused with toxic protective chemicals, resins, tannins, and oils that make it resistant to decay and to attack by insects and fungi. It is the tree's fortress, its historical core, a vast physical library of its own past. The evolution of the tree had a profound and lasting impact on the planet. The first great forests of the Carboniferous period changed the world's climate. Through the relentless work of photosynthesis, these vast new forests drew down immense quantities of carbon dioxide from the atmosphere, sequestering it in the form of wood. This led to a period of global cooling, and also to a dramatic rise in the oxygen content of the atmosphere, which may have allowed for the evolution of the giant insects of that era, like the dragonfly with a 2-foot wingspan. When these great Carboniferous trees died, they fell into the swampy, low-oxygen environments in which they grew. The normal processes of decay were inhibited, and over millions of years, the immense carbon-rich biomass of these first forests was buried, compressed, and cooked by the Earth's heat to be transformed into the vast deposits of coal that would one day power our own industrial revolution. The sunlight that fell on a Devonian forest 350 million years ago was captured, converted into wood and stored underground to be released by us in a geological instant as the energy that built our modern world. Every time we burn a piece of coal, we are releasing the ghost of an ancient wooden skyscraper. To contemplate the rise of the tree is to meditate on the power of a single biological innovation to change the face of the planet. Wood was the invention that allowed life to conquer the sky. It created a new, three-dimensional habitat, a complex, multi-layered world of canopy and understory that would become home to countless new species of animals and insects. The tree is a symbol of strength, of patience, and of a deep, quiet connection to the earth and the sky. It is a living bridge between two worlds, a silent wooden testament to the relentless, creative, and skyward ambition of the plant kingdom. The invention of wood gave plants the structural strength to grow tall, to build their magnificent wooden skyscrapers, and to reach for the sun. But this new, vertical dimension created a profound and daunting logistical challenge. A tall tree is an organism that is living in two different worlds at once. Its leaves, the photosynthetic factories, are high up in the dry sunlit air. Its roots, the absorbing organs, are deep down in the dark, moist soil. These two essential parts of the plant can be separated by tens or even hundreds of feet. The tree had to evolve a way to connect these two worlds. It needed a transportation system, a circulatory system, that could move vast quantities of water and minerals from the roots up to the leaves, and at the same time move the energy-rich sugars produced in the leaves back down to feed the trunk and the roots. This is the story of the plant's silent, powerful, and beautiful circulatory system, the story of a quiet, constant river flowing to the sky. Let us follow the journey of a single water molecule. Our journey begins in the soil, in the thin film of moisture that clings to a particle of clay. The water molecule is drawn, by the process of osmosis, across the delicate membrane of a root hair, into the body of the plant. From there, it travels from cell to cell, moving towards the center of the root, until it reaches a specialized star-shaped core of tissue. This is the xylem. The xylem is the plant's water-conducting pipeline. It is a network of tiny, hollow tubes, the dead, lignified remnants of once-living cells that form a continuous, unbroken plumbing system, stretching from the tip of the finest root, up through the trunk and the branches, to every single leaf in the canopy. Once the water molecule enters the xylem in the root, it begins a remarkable, upward journey, a journey that seems to defy the fundamental law of gravity. In a tall redwood or eucalyptus tree, this journey can be over 300 feet, the equivalent of a 30-story building. How does a plant with no heart, no pump, no moving parts, manage to lift a column of water to such a staggering height? For centuries, this was one of the greatest mysteries in botany. The answer lies not in a single powerful mechanism, but in a beautiful and subtle interplay of physical forces. The first small push comes from the roots themselves. The active pumping of mineral ions into the root xylem creates a high solute concentration, which draws water in through osmosis, creating a positive root pressure that can push the water column up the stem for a short distance. We can sometimes see the result of this root pressure in the early morning, when small droplets of water are forced out of the tips of the leaves, a phenomenon known as guttation. But root pressure is a weak force. It can only account for pushing the water up a few feet at most. It cannot explain how water reaches the top of a tall tree. The true engine that drives this river to the sky is not in the roots, but high above, in the leaves, and the force it uses is not a push from below but a pull from above. This mechanism is known as the cohesion tension theory. It is a process that is powered ultimately by the energy of the sun. Let us return to our water molecule, which has now travelled up the xylem and into the intricate network of veins within a leaf. The leaf is a moist, spongy environment. Its internal airspace is saturated with water vapour. On the underside of the leaf are the thousands of tiny pores, the stomata, that the plant uses to breathe in carbon dioxide. When these stomata are open, water vapour from inside the leaf diffuses out into the drier, outside air. This process is called transpiration. It is, in essence, the plant's form of sweating. This transpiration, this constant evaporation of water from the surface of the leaves, is the engine of the entire system. As a water molecule evaporates from the surface of a cell inside the leaf, it creates a tiny, microscopic tension, a negative pressure, at the air-water interface. This tension pulls on the next water molecule in the chain, which in turn pulls on the next, and the next. This pull is transmitted all the way down the continuous unbroken column of water in the xylem, from the leaf, down the branch, down the trunk and into the root, where it pulls another water molecule in from the soil to replace the one that was lost. This mechanism is only possible because of the remarkable physical properties of water itself. Water molecules are polar. They have a slight positive charge on one side and a slight negative charge on the other. This causes them to be strongly attracted to each other, a force called cohesion. It is this cohesion that allows the water molecules to stick together in a long, unbroken chain, like tiny magnets, allowing the tension from the top to be transmitted all the way to the bottom. The water molecules are also attracted to the walls of the narrow xylem tubes, a force called adhesion, which helps to support the water column and prevent it from breaking. The entire xylem system operates like a giant passive drinking straw. The evaporation from the leaves is the suction, and the cohesive and adhesive properties of water ensure that the straw never empties. The sheer scale of this process is staggering. A single large oak tree can transpire over 100 gallons of water on a hot sunny day. A forest is a vast, silent biological machine that is constantly drawing water up from the ground and releasing it into the atmosphere, shaping local and even global weather patterns. This process seems incredibly wasteful. The plant loses over 95% of the water it absorbs from the soil to transpiration. But it is a necessary cost of doing business. It is the unavoidable consequence of opening its stomata to take in the carbon dioxide it needs for photosynthesis. Transpiration is the price the plant pays for its food, but it also serves other vital functions. The evaporation of water from the leaves has a powerful cooling effect, preventing the leaf from overheating in the direct sun. And the constant flow of the transpiration stream is what carries the essential mineral nutrients, absorbed by the roots, up to the leaves where they are needed. While the xylem is responsible for the upward flow of water, the plant has a second parallel circulatory system for downward transport. This is the phloem. The phloem is a network of living cells that transports the energy-rich sugars produced during photosynthesis in the leaves to all the other parts of the plant that cannot photosynthesize for themselves: the trunk, the roots, the flowers, and the fruits. Unlike the passive, tension-driven flow in the xylem, the movement of sap in the phloem is an active, energy-intensive process. The plant actively loads the sugar into the phloem cells in the leaf, creating a high concentration. This draws water in from the nearby xylem through osmosis, creating a high pressure that pushes the sugary sap down the phloem, from the source, the leaf, to the sink, the root or the fruit, where the sugar is unloaded and used for energy or growth. To contemplate the circulatory system of a plant is to appreciate the silent, hidden and powerful currents that are constantly flowing within the green world. It is a system of beautiful physical elegance, a system that uses the simple properties of water and the energy of the sun to perform the seemingly impossible task of lifting a river to the sky. It is a perfect and efficient solution to the fundamental challenge of being a tall terrestrial organism. It is the intricate internal plumbing that knits the two worlds of the plant, the world of the root and the world of the leaf, into a single, unified and living whole. It is the quiet, life-giving pulse of the silent kingdom. Let us now journey to the end of the great upward-flowing river of xylem, to the place where the water, drawn up from the dark earth, finally meets the light of the sun. Let us contemplate the organ that is the ultimate destination of this journey, the engine of the entire plant, and, in a very real sense, the power source for almost all of life on Earth. This is the leaf. We are so accustomed to the sight of leaves that we often fail to appreciate the sheer, breathtaking elegance and efficiency of their design. A leaf is not just a simple, green appendage. It is a perfect, miniature, biological machine, a living solar panel that has been exquisitely optimized by hundreds of millions of years of evolution for a single, vital purpose: to capture the ethereal energy of sunlight and to transform it into the solid, chemical currency of life. To truly appreciate the genius of the leaf, we must look at it not as a static object, but as a solution to a complex set of engineering problems. The first problem is to maximize the capture of sunlight. A leaf's most obvious feature, its broad, flat shape, is a direct and elegant solution to this problem. It is a design that creates the largest possible surface area for intercepting photons, the tiny packets of light energy that rain down from the sky. The arrangement of leaves on a plant, a field of study known as phyllotaxy, is also a marvel of mathematical optimization. Leaves are typically arranged in a spiral pattern around the stem, with the angle between successive leaves often approaching the golden angle of 137.5 degrees. This precise geometric arrangement ensures that the leaves shade each other as little as possible, allowing the entire plant to act as a perfectly structured three-dimensional solar array. Let us now zoom in and look at the internal structure of the leaf, a hidden world of intricate and specialized tissues. The outer surface of the leaf is protected by a thin, transparent layer of cells called the epidermis, which is itself coated with the waxy, waterproof cuticle that we have encountered before. This transparent skin allows sunlight to pass through unhindered to the photosynthetic cells below. Scattered across the epidermis, particularly on the cooler, shaded underside of the leaf, are the thousands of tiny adjustable pores, the stomata. These are the gateways to the leaf, the microscopic mouths that allow the leaf to breathe in the carbon dioxide it needs from the atmosphere. Beneath the upper epidermis lies the heart of the leaf's solar-powered factory, the mesophyll. The mesophyll is divided into two distinct layers. The upper layer is the palisade mesophyll. It is a tightly packed, orderly arrangement of elongated, column-shaped cells, like a dense forest of green pillars. These cells are packed to the brim with the tiny green organelles that are the actual sites of photosynthesis, the chloroplasts. The orderly, vertical arrangement of the palisade cells is a clever design that allows the sunlight to penetrate deep into the leaf, passing through several layers of cells and maximizing the chance that every photon will be captured. Beneath the palisade layer is the spongy mesophyll. Here, the cells are irregular in shape and are loosely packed, creating a vast network of interconnected airspaces, like a porous, green sponge. This network of airspaces is connected to the outside world via the stomata. This structure is another masterpiece of design. It creates a huge, moist, internal surface area that allows for the efficient diffusion of gases. Carbon dioxide entering through the stomata can quickly and easily travel through these airspaces to reach every single photosynthetic cell, and the oxygen that is produced as a waste product of photosynthesis can just as easily diffuse out. The chloroplasts themselves are a world within a world. Each one is a tiny, self-contained, solar-powered factory, a descendant of the ancient cyanobacteria that first invented photosynthesis billions of years ago. Inside the chloroplast is a complex system of internal membranes, stacked up like tiny piles of green coins. These are the thylakoids, and it is in the membranes of these thylakoids that the chlorophyll molecules are embedded and where the first, light-dependent reactions of photosynthesis take place. Here, the energy of sunlight is captured and used to split water molecules, releasing oxygen and creating two high-energy molecules, ATP and NADPH. These are the short-term chemical batteries that will power the next stage of the process. The second stage, the light-independent reactions, takes place in the fluid-filled space surrounding the thylakoids, an area called the stroma. Here, in a complex cyclical chemical pathway known as the Calvin cycle, The energy from the ATP and NADPH is used to perform the final act of chemical alchemy. The cell takes the carbon dioxide it has breathed in from the air and, through a series of enzyme-catalyzed reactions, fixes it. It transforms the inorganic carbon of the atmosphere into the organic carbon of a simple 3-carbon sugar. This sugar is the fundamental product of photosynthesis. It is the food that the plant has created from light, air, and water. This sugar is the source of all of the plant's energy and all of its physical substance. It can be burned for immediate energy in the process of cellular respiration. It can be linked together to form cellulose, the long fibrous polymer that makes up the plant's cell walls and gives it its structural strength. It can be converted into starch, a more compact molecule for long-term energy storage in the roots or the seeds, or it can be used as the chemical backbone to build all the other complex organic molecules the plant needs, the proteins, the lipids, the vitamins, and the DNA. The leaf is not just a solar panel. It is a complete self-sustaining factory, capable of manufacturing everything it needs from the simplest of raw materials. But the life of a leaf is a constant and delicate balancing act. It is a series of compromises between competing needs. The leaf needs to be open to the air to get carbon dioxide, but every moment its stomata are open, it is losing precious water to transpiration. The plant must constantly, second by second, adjust the opening and closing of its stomata to find the optimal balance between carbon gain and water loss. This is a dynamic and highly regulated process, controlled by a complex interplay of signals, including the availability of light, the concentration of carbon dioxide inside the leaf, and the overall water status of the plant. The leaf must also protect itself. A leaf is a rich and nutritious package of energy, and it is a prime target for a vast array of herbivores, from the smallest insect to the largest mammal. Plants have evolved a staggering arsenal of defenses to protect their precious solar panels. Some have physical defenses, like tough leathery cuticles, sharp spines, or tiny hair-like trichomes that can make it difficult for an insect to walk on or chew the leaf. Others have chemical defenses. They produce a vast and complex array of toxic or bad-tasting compounds, alkaloids, tannins, cyanides, that can sicken or kill an aspiring herbivore. A leaf is not a passive meal. It is a well-defended fortress. To contemplate the leaf is to be filled with a sense of wonder at the elegance and efficiency of nature's design. It is a structure that is at once simple and profoundly complex. It is a solar panel, a chemical factory, a lung, and a fortress, all rolled into one thin green package. It is the point of connection between the inorganic and the organic, the place where the energy of a distant star is woven into the living fabric of our world. Every time we take a breath, we are breathing in the oxygen that was released as a gift from a leaf. Every bite of food we eat, whether it is a fruit, a vegetable, or the meat of an animal that ate plants, is ultimately a gift of stored sunlight, a gift from a leaf. The leaf is the silent, tireless, and life-giving engine of our planet, the beautiful green machine that powers our world. For hundreds of millions of years, the plant world was a world of silent, stoic green. The great forests of the Carboniferous period were vast, monotonous, and flowerless. The plants of this era, the ferns and the club mosses, reproduced through simple, inconspicuous spores, casting their genetic dust to the wind and hoping, by sheer force of numbers, that some of it would land in a suitable place to grow. It was a world without the vibrant colour of a petal, and it was a world without the complex, evocative magic of a flower's scent. The story of the flower and of its scent is the story of a revolutionary new strategy for reproduction, a strategy that was based not on the brute force of the wind, but on the subtle and seductive art of communication. It is the story of how plants learned to speak, not with sounds, but with a rich and complex language of chemicals, a fragrant message sent out upon the wind. The first flowers appeared relatively late in the history of plant life during the Cretaceous period, the age of the dinosaurs. They were likely small, simple, and inconspicuous structures, perhaps something like a modern magnolia, but they represented a profound evolutionary leap. The flower was a new and highly specialized reproductive organ, a structure designed not just to produce pollen and seeds, but to actively solicit the help of a third party in the process of pollination. That third party was the animal kingdom, particularly the burgeoning world of the insects. The wind as a pollinator is powerful, but it is incredibly inefficient and imprecise. For every grain of pollen that by pure chance lands on a receptive female cone, Millions or billions are wasted, falling to the ground or landing on the wrong species. It is a strategy of brute force and immense waste. The flower was a solution to this problem. It was a way to recruit a dedicated, reliable, and highly targeted delivery service. An insect like a bee or a beetle could be tempted to visit a flower, and in the process, it would inadvertently pick up a small, precise cargo of pollen and deliver it directly to the next flower it visited. This was a far more efficient and targeted system, but to make it work, the plant had to solve a new problem: how to attract the insect. The first and most obvious solution was to offer a reward. Most flowers produce a small quantity of a sugary, energy-rich liquid called nectar. For an active flying insect, this is a valuable source of food. The flower is, in essence, paying the insect for its delivery services, but a reward is useless if the potential customer doesn't know it's there. The plant needed a form of advertising. It needed a way to announce from a distance, "I have a reward for you. Come here." Visual signals were one answer. The evolution of the petal, a modified leaf, provided a colourful billboard. The vibrant reds, yellows, and blues of a flower's petals are a stark and beautiful contrast to the uniform green of the foliage, making the flower stand out in the landscape. Many flowers also evolved intricate patterns on their petals, called nectar guides, which are often visible only in the ultraviolet spectrum of light that insects can see. These patterns act like the landing lights on an airport runway, guiding the insect directly to the source of the nectar and ensuring that it will brush up against the flower's reproductive organs, the stamens and the pistil, in the process. But the most subtle, the most ancient, and perhaps the most powerful form of floral advertising is the scent. A flower's fragrance is not a simple, accidental perfume. It is a complex and carefully crafted chemical message, a plume of volatile organic compounds that is released into the air and carried on the wind, often for great distances. It is a long-distance advertisement, capable of reaching its target audience long before the flower is even visible. Let us contemplate the chemical complexity of a single floral scent. The fragrance of a rose, for example, is not a single molecule. It is a complex cocktail of hundreds of different volatile compounds, alcohols, esters, terpenes, all blended together in a specific and unique ratio. Each of these individual compounds may be produced by other plants, but the specific combination, the unique recipe, is the signature of the rose. It is this complexity that allows for the incredible diversity of floral scents in the natural world, from the sweet, heady perfume of a jasmine to the spicy clove-like scent of a carnation, to the strange, fetid odour of the carrion flower, which mimics the smell of rotting meat to attract its preferred pollinators, flies and beetles. The plant's control over its scent is incredibly precise. It is not releasing this fragrant message all the time. That would be a waste of energy. The production and release of scent is often tightly regulated, timed to coincide with the activity of its target pollinator. A flower that is pollinated by bees or butterflies will release its scent during the day, but a flower that is pollinated by nocturnal moths or bats, like the night-blooming jasmine or the cereus cactus, will wait until dusk. As the sun sets and the world grows dark, it will open its pale, luminous petals and release its powerful, intoxicating perfume into the cool night air, a fragrant beacon in the darkness. The relationship between a flower and its pollinator is one of the most beautiful examples of coevolution in the natural world. Over millions of years, the flower and the pollinator have shaped each other. The flower has evolved a scent, a colour, and a shape that is perfectly tailored to the senses and the body of its preferred partner, and the pollinator has evolved the sensory equipment and the behaviour to find and to exploit its floral partner. Some of these relationships are highly specialised. The scent of a specific orchid, for example, may mimic the exact chemical composition of the sex pheromone of a particular species of female wasp. The male wasp, thinking he has found a mate, will try to copulate with the flower, and in the process he will inadvertently pollinate it. The orchid has evolved a form of chemical deception, a fragrant lie that ensures its own reproductive success. But the language of scent is not just used for attraction, it is also used for defense. When a plant's leaves are being eaten by a caterpillar, the plant doesn't just suffer in silence, it changes the chemical message it is sending. It begins to release a new and different blend of volatile compounds, a chemical scream for help. This new scent is a distress signal, and it can have several effects. It can act as a warning to other nearby plants, signaling them to ramp up their own chemical defences in preparation for an attack. And, in a truly remarkable feat of inter-kingdom communication, this distress signal can act as a beacon for predatory insects, like parasitic wasps. The wasp will follow the scent trail back to the plant, find the caterpillar that is causing the damage, and lay its eggs inside it. The plant under attack has effectively called in an airstrike on its enemy. To contemplate the scent of a flower is to enter a world of hidden chemical conversations. It is to understand that the air around us, which we perceive as empty, is in fact filled with a constant, complex and meaningful stream of information. A flower's fragrance is a message, a story, a seduction, and a warning. It is a testament to the profound and subtle ways in which plants interact with the world around them. It is a reminder that communication is not limited to the sounds and sights of the animal kingdom. The silent world of plants is, in fact, a world that is constantly, quietly, and fragrantly talking. The evolution of the flower, with its vibrant colours and its seductive scents, was a profound and beautiful invitation. It was a call sent out from the silent, stationary world of the plant kingdom to the active, mobile world of the animal kingdom. It was an invitation to participate in one of the most intimate and important acts of a plant's life, the act of pollination. Pollination is the transfer of pollen, the male genetic material, from the anther of one flower to the stigma of another. It is the essential prelude to fertilization and the creation of a seed. The story of pollination is the story of how plants, bound to the earth, solved the problem of finding a mate. And it is the story of the birth of one of the most intricate, beautiful, and important partnerships in the history of life on Earth, the co-evolutionary dance between the flower and the animal. For hundreds of millions of years, the primary agent of pollination was the wind. The great coniferous forests of the Mesozoic era reproduced by casting vast profligate clouds of pollen into the air, relying on pure statistical chance to carry a few of those grains to their destination. This is a strategy of immense waste. The vast majority of the pollen is lost, and the plant must invest a huge amount of energy in producing these astronomical quantities. The evolution of animal pollination was a revolutionary leap in efficiency and precision. By recruiting an animal to act as a dedicated courier, the plant could produce far less pollen and have a much higher certainty that it would arrive at its intended target. The animal, in effect, became a mobile, external reproductive organ for the plant. This new strategy required two things: a way to attract the animal and a way to reward it. We have seen how the flower evolved its advertisements, the colours and the scents. The reward was typically nectar, a rich, sugary liquid that provided a high-energy food source for an active, flying animal. But the flower also had to evolve a physical structure, a morphology, that would facilitate the transfer of pollen. The architecture of a flower is not accidental. It is a carefully designed landing pad, a machine for pollination. Let us contemplate the intricate dance that takes place between a bee and a flower. The bee, guided by the flower's colour and scent, lands on a petal. To get to the nectar, which is often located deep within the base of the flower, the bee must push its way past the flower's reproductive organs. As it does so, its fuzzy body brushes up against the anthers, the male organs, which are laden with sticky pollen grains. The pollen adheres to the bee's body. The bee drinks its fill of nectar and then flies off, its body now dusted with a fine yellow powder. When it arrives at the next flower of the same species, the dance is repeated, but this time, as the bee pushes its way towards the nectar, some of the pollen from its body is transferred onto the flower's stigma, the sticky, receptive female organ. Pollination is complete. The bee has been paid for its services, and the flower has achieved its reproductive goal. This is a relationship of mutualism, a partnership from which both parties benefit. The bee gets a reliable source of food, and the flower gets a reliable and highly efficient pollination service. Over millions of years, this relationship has become more and more specialized. The flower has evolved to be a perfect fit for the bee, and the bee has evolved to be a perfect pollinator for the flower. The length of a flower's corolla tube may evolve to be a perfect match for the length of a bee's tongue. The flower may evolve a specific landing platform, a petal that is just the right size and shape for the bee to land on. The bee, in turn, may evolve a preference for a specific colour or scent, and it may learn to visit only one type of flower on any given foraging trip, a behaviour known as flower constancy. This is highly advantageous for the plant, as it ensures that its precious pollen is not wasted by being delivered to the wrong species. The bee is perhaps the most famous and important of all pollinators, but it is by no means the only one. The plant kingdom has recruited a vast and diverse army of animal partners. Butterflies, with their long, slender tongues, are important pollinators for flowers that have a deep, narrow floral tube. Moths, which are active at night, are attracted to flowers that are pale or white, making them more visible in the moonlight, and that release their scent only after dusk. The hawk moth, with its incredible hovering flight, is a key pollinator for flowers like the honeysuckle, which have a long dangling structure. Birds, particularly hummingbirds, are another important group of pollinators. Hummingbirds have a very poor sense of smell, but they have excellent colour vision, and they are particularly attracted to the colour red. And so, the flowers that seek to attract hummingbirds have co-evolved with them. They are typically red or orange, they have little to no scent, and they produce a large quantity of a more watery nectar, suited to the high metabolism of the bird. They are also often shaped like a long hanging tube, perfectly designed for the hummingbird to insert its long, slender beak while it hovers in mid-air. Even mammals have been recruited into this great pollination dance. Bats are crucial pollinators in many tropical and desert ecosystems. Like moths, they are active at night, and so the flowers that attract them are also typically pale, large, and open only after dark. But bat-pollinated flowers have a different kind of scent. It is not a sweet floral perfume, but a strong, musky and almost fermented odour, a scent that is attractive to a bat. They also produce a huge amount of nectar to reward their large, high-energy pollinator. In some parts of the world, other non-flying mammals like mice, lemurs and even possums have also been found to be important pollinators for certain ground-dwelling plants. But the relationship between the plant and the animal is not always one of honest mutual benefit. Sometimes it is a relationship of deception and trickery. We have already seen how some orchids mimic the scent and appearance of a female insect to trick the male into pollinating them, a form of sexual deception. This is a purely exploitative relationship. The orchid gets pollinated, but the insect gets nothing in return for its efforts. There are also nectar robbers. These are animals, often bees or birds with short tongues, that have figured out a way to cheat the system. They cannot reach the nectar through the front of a long tubular flower, so they simply chew a hole in the base of the flower and steal the nectar without ever coming into contact with the pollen. The plant has been robbed, it has lost its precious energy-rich reward, and it has received no pollination service in return. The Great Pollination Dance is a beautiful and intricate web of relationships, a complex interplay of cooperation and competition, of honesty and deception. It is a story that is written in the shape of a flower's petal, in the colour of its bloom, and in the chemical language of its scent. It is a story that highlights the profound interconnectedness of the living world, the way in which two entirely different kingdoms of life, the plant and the animal, have become deeply and inextricably dependent on one another. This ancient partnership is now under threat. The widespread use of pesticides, the destruction of natural habitats, and the effects of climate change are all contributing to a dramatic and alarming decline in the populations of many of our most important pollinators, particularly the bees. This is not just a tragedy for the bees themselves, it is a profound threat to the health of our ecosystems and to the security of our own food supply. A huge proportion of the food crops that we depend on, from fruits and vegetables to nuts and coffee, are reliant on animal pollination. The silent, beautiful and ancient dance between the flower and the animal is a dance that sustains us all. To contemplate this great pollination is to be reminded of the delicate, intricate and often invisible threads that hold our world together, and of our own profound responsibility to protect them. The dance of pollination is complete. A grain of pollen has been successfully delivered to the stigma of a receptive flower. This is the moment of conception. The pollen grain grows a tiny tube down through the style of the flower, until it reaches the ovule, hidden deep within the flower's ovary. The male genetic material travels down this tube and fuses with the female egg. Fertilization has occurred. A new life, a tiny embryonic plant, has been created. But the plant's reproductive journey is far from over. This new vulnerable embryo must be protected, it must be nourished, and it must be sent out into the world to find its own place to grow. The plant's solution to this challenge is one of the most remarkable and beautiful inventions in the entire history of life. It is the invention of the seed. A seed is not an inert object. It is not a speck of dust. It is a living, breathing, and incredibly sophisticated time capsule. It is a tiny, sleeping baby plant, a complete organism in miniature that has been carefully packaged with everything it will need to begin its life, and then put into a state of suspended animation, waiting patiently for the perfect moment to awaken. To contemplate a seed is to hold an entire potential future in the palm of your hand. It is to witness a masterpiece of biological engineering, a vessel of time, and a profound symbol of hope and resilience. Let us look at the intricate structure of a typical seed. At its heart is the embryo itself. The embryo is a complete miniature plant, already possessing the rudimentary structures that will become its future roots, its stem, and its first leaves. It is a tiny, dormant blueprint of the mature plant. Surrounding the embryo is its food supply, the endosperm. This is a starchy, nutrient-rich tissue, packed with all the energy and the raw materials the embryo will need to fuel its initial growth when it finally awakens. It is the packed lunch that the mother plant has provided for its offspring's long journey, and enclosing both the embryo and the endosperm is the seed coat. This is the protective outer shell of the seed, a tough, durable, and often waterproof layer that shields the precious cargo within from physical damage, from drying out, and from being eaten by predators. The most remarkable property of the seed is its ability to wait. The seed is a master of patience. Once it has been formed and has matured on the parent plant, it undergoes a process of profound desiccation. It loses almost all of its water, and its metabolic activity slows down to an almost undetectable level. It enters a state of deep dormancy, a state of suspended animation that is closer to hibernation than to life as we normally know it. In this dormant state, a seed can survive for incredible lengths of time, waiting for the precise set of environmental cues that will signal that the time is right to germinate. The length of this waiting period can be astonishing. While the seeds of many plants will germinate in the next growing season, some can remain viable in the soil for decades, or even centuries, forming a seed bank, a hidden living memory of the past vegetation of a place. In one famous experiment, scientists took soil from a field that had been continuously farmed for over 100 years, and found that it still contained viable seeds of the weeds that had grown there a century before. When given the right conditions, these ancient seeds sprouted into life. The most dramatic example of this longevity comes from a 2,000-year-old date palm seed that was discovered during an archaeological excavation at the ancient fortress of Masada in Israel. In 2005, scientists planted this ancient seed, and to their astonishment, it germinated. A plant that was born from a seed that was created during the time of the Roman Empire is now growing in the 21st century. The seed is a bridge across the millennia. What is the seed waiting for? Each species has its own, unique set of germination requirements, a secret environmental password that must be entered before the seed will awaken. For many seeds, the primary requirement is the presence of water. When the seed absorbs water, it swells, and this triggers the metabolic processes that will break its dormancy. But for many seeds, water alone is not enough. They are waiting for other, more specific cues. Some seeds require a period of prolonged cold, a process called stratification. This is a clever adaptation that prevents the seed from germinating during a warm spell in the autumn, only to be killed by the coming winter. The seed must experience the cold of winter before it will be ready to sprout in the spring. We have contemplated the visible world of the plant, the intricate architecture of the leaf and the flower, and the towering strength of the wooden trunk. But to truly understand the secret life of a plant, we must journey underground, into the dark, silent, and hidden world of the roots. We have seen the root as an anchor, and as a mouth, a tool for stability and for nourishment. But in the last few decades, a revolutionary new understanding has emerged, an understanding that has completely transformed our view of the forest. We have discovered that the root systems of individual trees are not isolated and separate. They are in fact connected to each other, woven together into a vast, intricate, and ancient network, a biological internet that lies hidden beneath the forest floor. This is the story of the Wood Wide Web, a story of a profound and secret partnership. a story that reveals the forest not as a collection of individual trees, but as a single, vast, interconnected, and social superorganism. The architects of this underground network are not the plants themselves, but their silent and ancient partners, the mycorrhizal fungi. The word mycorrhiza literally means fungus root. It refers to the symbiotic, mutually beneficial relationship that exists between the roots of most plants and a specific group of soil fungi. This is not a rare or unusual relationship, it is the norm. Over 90% of all plant species on Earth form these mycorrhizal associations. It is a partnership that is as old as the land plants themselves, a partnership that likely began over 450 million years ago and was essential for the first plants to even survive on the harsh, primitive land. Let us journey into the soil and witness this partnership. A fungal spore, a tiny microscopic speck, germinates in the soil near a plant's root. It sends out a fine, thread-like filament called a hypha. This hypha grows towards the root, and when it makes contact, it begins to interact with it. There are two main types of mycorrhizal relationships. In one, the ectomycorrhiza, the fungal hyphae form a dense, thick sheath around the outside of the root tip, and then grow in a net-like pattern between the root cells, but without actually penetrating them. In the other, more common type, the arbuscular mycorrhiza, the fungal hyphae actually penetrate into the root cells themselves. Once inside the cell, the hypha branches out into a beautiful, intricate tree-like structure called an arbuscule. This is the site of the exchange, the marketplace where the plant and the fungus trade their goods. It is a simple and elegant bargain. The plant, through the process of photosynthesis in its leaves, is able to produce an abundance of energy-rich sugars, but it often struggles to absorb enough of the essential, but scarce, mineral nutrients from the soil, particularly phosphorus and nitrogen. The fungus, on the other hand, is a master of absorption. Its vast, sprawling network of hyphae, which are far, far thinner than even the finest root hair, can explore a huge volume of soil with an incredible efficiency. The fungus is brilliant at scavenging for and absorbing phosphorus and nitrogen, but it cannot produce its own food, it cannot photosynthesize, and so the deal is struck. At the site of the arbuscule, the plant gives the fungus the sugars it needs to live and to grow. It can divert as much as 20% to 30% of its total photosynthetic production to feeding its fungal partner. In return, the fungus acts as a vast extended root system for the plant. It explores the soil, absorbs water and mineral nutrients, and transports them back to the root, delivering them directly into the plant's cells. The fungal network is so much more efficient at this than the plant's own roots that a plant with a mycorrhizal partner is significantly healthier, more robust, and more resilient than one without. The fungus is, in essence, an external digestive system for the plant, breaking down complex organic matter in the soil and making the nutrients available. It also protects the plant from root diseases and from toxic heavy metals. But the story becomes even more profound when we zoom out. The network of a single fungus does not just connect to a single plant. The vast underground web of fungal hyphae, the mycelium, can be enormous, stretching for miles, and it can connect to the roots of many different plants, even plants of different species. A single ancient fungus can be physically connected to hundreds of trees in a forest. And so, the individual trees of the forest, which appear to us to be separate and competing individuals, are in fact all plugged into the same shared underground network. The forest floor is not a collection of separate root systems. It is a single, integrated biological internet. This wood wide web is not just a passive plumbing system for the delivery of nutrients. It is a dynamic and active communication network. It is a channel through which the trees of the forest can talk to each other. We are just beginning to decipher the complex conversations that are taking place in this hidden subterranean world. We have learned that trees can use this network to share resources. A large, healthy mother tree, a hub in the network with access to abundant sunlight, can send excess carbon and nutrients through the fungal network to smaller, younger trees that are struggling in the shade of the understory. The mother tree is, in a sense, nurturing its own offspring, and the offspring of its neighbours, giving them the resources they need to survive until they can reach the canopy themselves. This is not a world of pure, ruthless competition. It is a world of cooperation, of redistribution, and of community. The network is also a conduit for information. When a tree is attacked by an insect herbivore, it can send a chemical distress signal through the fungal network to its neighbours. The neighbouring trees, upon receiving this signal, can ramp up their own defensive chemistry, preparing for the attack before the insect even arrives. The fungal network acts as a forest-wide alarm system, a silent underground telegraph, allowing the trees to mount a coordinated community-wide defence. This underground network can also be a place of competition and of warfare. Some plants have learned to hack the system. There are certain non-photosynthetic plants, like the strange and beautiful ghost pipe, that are pure parasites on the network. They produce no chlorophyll and make no food of their own. Instead, they plug their roots into the mycorrhizal network and simply steal the sugars that are being passed between the trees and the fungi. Other, more aggressive plants can use the network to wage a form of chemical warfare, sending toxic allelopathic chemicals through the fungal hyphae to inhibit the growth of their rivals. The discovery of the wood wide web is a profound paradigm shift in our understanding of a forest. It forces us to see the forest not as a collection of individual competing entities, but as a complex adaptive system, a superorganism. The trees, the fungi, the soil, the microbes, they are all part of a single, deeply interconnected and co-evolved whole. The health of the individual is inseparable from the health of the community. The intelligence of the forest is not located in any single tree, but is distributed throughout the entire network. It is a form of swarm intelligence, a collective consciousness that emerges from the simple, local interactions of millions of individual agents. To contemplate this underground web is to be filled with a sense of wonder at the hidden complexity and the secret social life of the plant kingdom. It is a powerful reminder that the most important things in an ecosystem are often the things that are invisible to our eyes. It challenges our deeply ingrained individualistic view of the world and invites us to see the profound and beautiful truth of interconnectedness. The trees in a forest are not standing in isolation. They are holding hands underground. They are linked in a silent, ancient, and deeply supportive embrace, a community that is bound together by a vast living and intelligent web of fungal threads. For most of our history, we have seen plants as silent, passive, and solitary beings. They do not have mouths to speak, ears to hear, or nerves to carry signals. They are, to our animal-centric way of thinking, deaf, dumb, and alone. But this is a profound failure of our own imagination. The plant kingdom is not a silent world. It is in fact a world that is filled with a constant, complex, and deeply meaningful conversation. Plants are talking to each other and to the world around them all the time. They are just not using a language that we are accustomed to recognizing. Their language is not the language of sound or of sight, but the most ancient and universal language of all, the language of chemistry. Let us imagine a single plant, a bean plant, growing in a field. It is a peaceful scene. But then a threat appears. A small caterpillar begins to chew on one of its leaves. The plant is under attack. It is being wounded. But it does not suffer in silence. The moment its cells are damaged, it begins to respond. It initiates a complex and sophisticated series of defensive maneuvers, and a key part of this defense is communication. The first conversation is an internal one. The wounded leaf produces a cascade of chemical signals, plant hormones like jasmonic acid, that travel through the plant's own circulatory system. This is an internal alarm, a message that travels from the site of the wound to all the other, undamaged leaves on the same plant. The message says, "We are under attack. Prepare for battle." In response to this signal, the other leaves begin to produce their own defensive chemicals. They might start to produce toxic compounds that will sicken or kill the caterpillar if it moves to a new leaf. Or they might produce antifeedant compounds that make the leaves taste bitter and unpalatable. The plant is mounting a systemic body-wide defense, all coordinated by this internal chemical conversation. But the conversation does not stop there. The wounded plant also begins to talk to its neighbours. As the caterpillar chews on the leaf, the plant releases a specific and complex blend of volatile organic compounds, or VOCs, into the air. These are small, lightweight molecules that can travel on the wind. This is not just an accidental byproduct of the damage, it is a deliberate and meaningful signal. It is an airborne scream. A neighbouring bean plant, even one that is not yet under attack, has the ability to smell this chemical message. Its leaves have receptors that can detect these specific VOCs. When it receives this airborne warning signal from its wounded neighbour, It eavesdrops on the conversation. It understands the message, danger is near, and in response, it too begins to ramp up its own defensive chemistry. It prepares for the attack before the caterpillar has even arrived. The first plant, by releasing its chemical scream, has warned its community, allowing for a coordinated neighborhood-wide defense. This is a form of altruism, but it is also a form of self-interest. A healthy neighborhood is a safer neighborhood for everyone. But the conversation gets even more complex and even more astonishing. The chemical scream released by the wounded plant is not just a general alarm, it is a highly specific and information-rich message. The specific blend of VOCS that a plant releases can tell a very detailed story. It can indicate not just that it is being attacked, but who is attacking it. The chemical signature released in response to a caterpillar is different from the signature released in response to a fungal infection or a bacterial disease, and it can even be specific to the species of caterpillar. The plant is, in essence, shouting, "I am being eaten by a monarch butterfly caterpillar, and who is this specific message for?" Incredibly, it is often for a third party, an ally from the animal kingdom. The plant, under attack, is calling for a bodyguard. Let us return to our bean plant being eaten by a caterpillar. The specific blend of VOCS that it releases into the air is a powerful attractant for a tiny predatory insect, the parasitic wasp. The wasp is the mortal enemy of the caterpillar. The female wasp can smell this chemical distress signal from a great distance. She follows the fragrant trail back to its source, back to the wounded bean plant. She finds the caterpillar that is causing the damage, and she performs a gruesome but, for the plant, highly effective act. She lands on the caterpillar and injects her eggs inside its living body. The wasp larvae then hatch inside the caterpillar and begin to eat it from the inside out, eventually killing it. This is a stunningly sophisticated tritrophic interaction, a conversation that crosses the boundaries between kingdoms. The plant, unable to physically defend itself, has evolved the ability to summon the enemy of its enemy. It has recruited a mercenary, a tiny winged assassin, and it has paid for its services not with nectar, but with a highly specific and targeted stream of information. It has turned its chemical language into a weapon. This chemical language is not just used for defense, it is the language of the plant's entire social life. The roots of a plant are constantly releasing a complex cocktail of chemicals, called exudates, into the soil. This is a way of communicating with the vast community of microbes in the rhizosphere. The plant uses these chemical signals to cultivate and to talk to its beneficial partners, the mycorrhizal fungi and the nitrogen-fixing bacteria. It sends out signals that say, "I am here, I am a friend, I have sugar to trade," and the microbes respond with their own chemical signals, a conversation that leads to the formation of the symbiotic relationships that are so essential for the plant's health. The roots also use these chemical signals to communicate with other plants. They can sense the chemical exudates of their neighbours. This allows them to distinguish between kin, their own close relatives, and strangers. When a plant senses that its neighbour is a stranger, it will often adopt a more competitive strategy, growing its roots more aggressively to claim a larger share of the available resources. But when it senses that its neighbour is a close relative, it will often become more cooperative, restraining the growth of its roots to avoid overcrowding its own family. This kin recognition is a form of nepotism, a strategy that helps to ensure the success of its own genetic lineage. Even the process of germination can be a chemical conversation. Some parasitic plants, like the witchweed, have evolved a sinister form of chemical eavesdropping. Their seeds will lie dormant in the soil for years, waiting. They will only germinate when they detect the specific chemical signal, a strigolactone that is released by the roots of a nearby host plant, like a corn or a sorghum plant. The parasitic seed is listening for the chemical whisper of its victim before it will awaken and attack. To contemplate this chemical language is to completely reimagine the world of the plant. It is to see the plant not as a silent, solitary object, but as a dynamic, communicative and social being. It is constantly sensing its environment, it is constantly assessing threats and opportunities, and it is constantly talking. It is talking to itself, it is talking to its neighbours, it is talking to its microbial partners in the soil, and it is talking to the insects in the air. The air and the soil, which seem so empty to us, are in fact filled with a rich, complex and constant stream of chemical information. This is a form of intelligence. It is not the fast, centralized neural intelligence of an animal. It is a slow, decentralized, and distributed intelligence that is woven into the very fabric of the plant's being, into its genes, its metabolism, and its development. The plant is a master of organic chemistry. It is a chemical conversationalist of breathtaking sophistication. To learn to see the world in this way is to be humbled. It is to realize that the silence of the plant kingdom is an illusion, born of the limitations of our own senses. The world is, in fact, humming with a quiet, fragrant, and deeply meaningful conversation, a conversation that we are only just beginning to learn how to hear. Our human experience of the world is defined by our 5 senses. We see with our eyes, we hear with our ears, we smell with our nose, we taste with our tongue, and we feel with our skin. These senses are our windows onto reality, the channels through which we gather information about the world around us. Because plants do not have eyes, or ears, or noses, or tongues, or skin in the way that we do, we have long assumed that they are insensate, that they are living in a world devoid of any sensory experience. We have seen them as objects, incapable of perceiving the world in which they live. But this is a profound and arrogant mistake, a failure to recognize that life has evolved many different and equally valid ways of sensing the world. Plants are, in fact, incredibly sensitive beings. They're constantly monitoring their environment, gathering a rich and complex stream of information about the world. They can see, they can touch, they can taste, they can smell, and they can sense things that are entirely beyond our own limited human perception. Their world is a rich and vibrant sensory landscape. They are just not using the same organs that we are. Let us first contemplate the plant's sense of sight. A plant does not have eyes, but it can see. Its primary sense of sight is its ability to detect the presence, the direction, the intensity, the duration, and the quality of light. Light is the single most important resource for a plant, and its ability to perceive and to respond to light is essential for its survival. The entire body of the plant, from the tip of its shoot to the leaves and the stem, is, in a sense, a single, giant, distributed eye. The plant's retina is a suite of sophisticated photoreceptor proteins, each one tuned to a specific wavelength, a specific color of light. The most well-known of these photoreceptors are the phototropins. These proteins are sensitive to blue light, and they are responsible for the phenomenon of phototropism, the ability of a plant to bend and to grow towards a light source. When blue light strikes the phototropins in the tip of a plant's shoot, it triggers a chemical signal that travels down the shaded side of the stem. This signal, a plant hormone called auxin, causes the cells on the shaded side to elongate more rapidly than the cells on the sunny side. This differential growth causes the stem to bend, orienting the leaves towards the source of the light, a slow, graceful, and deliberate act of seeing. But the plant can see more than just the direction of the light. It can also see the quality of the light, and it uses this information to see the world around it. The plant has another, even more remarkable set of photoreceptors called phytochromes. Phytochromes are like a reversible biological light switch. They are sensitive to red light and to far red light, the light at the very edge of the visible spectrum. When a phytochrome molecule is struck by red light, which is abundant in direct sunlight, it is switched into an active state. When it is struck by far red light, which is more abundant in the shade, it is switched back into an inactive state. The plant uses the ratio of active to inactive phytochrome as a way of sensing its environment. A plant growing in the open sun is bathed in red light, and most of its phytochrome is in the active state. This tells the plant that it has plenty of light, and it can invest its energy in growing strong stems and large leaves. But if another taller plant grows nearby and begins to cast a shadow over it, the quality of the light changes. The chlorophyll in the taller plant's leaves absorbs most of the red light for photosynthesis, but it allows the far red light to pass through. And so, the plant in the shade is now bathed in a light that is rich in far red. This switches its phytochrome back to the inactive state. This is a clear and unambiguous signal to the plant. You are in the shade. You are being outcompeted. In response to this signal, the plant will trigger a shade avoidance response. It will stop investing in broad leaves and will instead pour all of its energy into rapid vertical growth, elongating its stem in a desperate attempt to grow out of the shadow and to reach the sun. The plant has, in a very real sense, seen its competitor, not by forming an image, but by sensing its specific spectral shadow. The plant also has a profound sense of touch. We can see this most clearly in the behaviour of a climbing plant, like a pea or a morning glory. As the growing tip of the vine sways in the breeze, it is performing a slow circular scanning movement called circumnutation. It is, in essence, searching for something to hold on to. When the tip of the vine touches an object, like a fence post or the stem of another plant, it can feel it. Specialized cells in its epidermis detect the physical contact. This triggers a response. The vine will begin to coil around the object, its growth pattern changing to form a tight, secure spiral. The plant has found a support that will allow it to continue its journey towards the sky. This sense of touch can be incredibly sensitive. The tiny tendrils of a pea plant can detect a wire that is only a fraction of a millimetre in diameter, and they can remember the contact. If you touch a tendril, it will begin to curl. If you then let it go, it will uncurl. But if you touch it repeatedly, it will learn that the stimulus is not a reliable support, and it will stop responding. It has formed a simple, cellular memory of the touch. The most dramatic example of this sense of touch is, of course, the Venus flytrap. The tiny trigger hairs on the surface of its lobes are incredibly sensitive mechanoreceptors. When an insect brushes against one of these hairs, it sends an electrical signal, an action potential, that is remarkably similar to the nerve impulses in our own bodies. But a single touch is not enough. This prevents the trap from closing in response to a random piece of debris or a raindrop. The plant requires a second touch, within about 20 seconds of the first. This second signal tells the plant that the object is the right size and is moving in the right way to be a living insect. Only then will the trap snap shut with incredible speed. The Venus flytrap is not just feeling, it is counting. The plant also has a sense of taste. This sense is located primarily in its roots. As we have seen, the root system is constantly tasting the chemical composition of the soil. It can detect the presence and the concentration of water, of dozens of different mineral nutrients, of salts, of toxins, and of the chemical signals released by other organisms. The root is a discerning and sophisticated palate. It can guide its growth towards a pocket of rich, fertile soil and away from a patch of toxic or saline soil. It is performing a slow, subterranean form of gourmet dining, constantly seeking out the most nutritious and avoiding the least palatable patches of its environment. And the plant has a sense of smell. Its ability to detect and to respond to the volatile organic compounds released by its neighbours is a clear and well-documented form of olfaction. A plant can smell the chemical scream of a wounded neighbour and can prepare its defences accordingly. It can smell the ripeness of its own fruit, releasing the hormone ethylene, a gaseous signal that coordinates the ripening process across the entire plant, and it can smell the presence of a potential host. The parasitic dodder vine, a plant with no leaves and no roots of its own, has been shown to smell its preferred host, like a tomato plant. It will actively grow towards the scent of the tomato, ignoring other, less desirable plants nearby. It is a silent botanical predator, sniffing out its victim on the wind. To contemplate the senses of a plant is to be humbled by the diversity of life's solutions. It is to realize that our own, human-centric view of the world is just one, limited perspective. Plants are not living in a dull, insensate world. They are living in a world that is rich with information, a world of subtle colours, of gentle touches, of complex tastes, and of fragrant chemical conversations. They are deeply and intimately aware of the world around them. They are seeing, feeling, tasting, and smelling all the time. They are just doing it with a different kind of body, a different kind of mind, and on a different kind of timescale. Theirs is a quiet, distributed, and deeply patient form of consciousness, a form of consciousness that is woven into the very fabric of their being. One of the most fundamental distinctions we make between plants and animals is the distinction between the stationary and the mobile. Animals, we believe, are active, motile beings. They run, they fly, they swim, they hunt. They move through the world. Plants, on the other hand, are passive, sessile, and immobile. They are rooted to the spot. They are the silent, unmoving furniture of the natural world. But this perception, like so many of our assumptions about the plant kingdom, is an illusion. It is an illusion that is born of the profound difference in the timescale on which we and the plants operate. Plants are moving, all the time. They are exploring their environment, they are foraging for resources, they are competing with their neighbours, and they are responding to threats. They are just doing it so slowly, so gracefully, and so deliberately, that to our own, fast-paced animal consciousness, it appears as no movement at all. To truly see the secret life of plants, we must learn to see the world in plant time. The invention of time-lapse photography has given us a magical window into this hidden world of plant movement. It allows us to compress the slow, patient, and almost imperceptible movements of a plant, which can take place over hours, days, or even weeks, into a few fleeting seconds of video. And in doing so, it reveals a world of incredible dynamism, of graceful beauty, and of purposeful, intelligent action. Let us watch, in time-lapse, the germination of a simple bean seed. We see the seed swell as it absorbs water. We see the root emerge, a pale, questing finger that probes the soil, twisting and turning as it navigates the subterranean world. Then we see the shoot emerge, a bent hooked structure, like a shepherd's crook. This hook, the hypocotyl, is a clever piece of engineering. It is the part of the stem that pushes its way up through the abrasive soil, protecting the delicate embryonic leaves which are tucked safely behind it. Once the hook breaks through the surface and senses the light, it straightens up, pulling the first two leaves, the cotyledons, up into the air and unfurling them like a pair of green wings. The plant has been born, and its first great movement is complete. Now let us watch the young plant as it grows. We see that it is not static. The leaves are constantly adjusting their position, their angle, and their orientation throughout the day. This is the process of heliotropism, or solar tracking. The leaves are following the sun as it moves across the sky, from east to west like a field of tiny green satellite dishes, constantly repositioning themselves to maximize their exposure to the life-giving light. At night, in some plants, we can see the leaves fold up, a sleep movement called nectinasty, which may help to conserve heat or to protect the leaf from nocturnal herbivores. The plant is in a constant, gentle and rhythmic motion, a slow daily dance with the sun. The most dramatic and beautiful of all plant movements is the movement of a climbing plant. Let us watch a morning glory vine as it begins its ascent. The growing tip of the vine does not grow in a straight line. It performs a slow, graceful and continuous circular movement, a process called circumnutation. The tip is literally scanning its environment, like a blind person's cane, sweeping through the space around it in a wide arc, searching for something to touch. This is not a random passive swaying in the wind. It is an active, internally driven and genetically programmed behavior. The vine is hunting. It is hunting for a support. When the circling tip of the vine finally makes contact with a solid object, a pole or the stem of another plant, we see its behavior change instantly. The sense of touch triggers a new kind of movement. The vine begins to wrap itself around the support, its growth pattern changing to form a tight, secure spiral. It has found an anchor, a ladder that will allow it to continue its journey upwards, towards the sun. As it continues to grow, it will send out new, searching tendrils, each one performing its own slow, circular dance, looking for the next handhold. The climbing plant is an explorer, a mountaineer, a slow-motion acrobat, constantly, actively and intelligently navigating the three-dimensional world. Even the flowers of a plant are in motion. We can watch, in time-lapse, the slow, beautiful, and almost sensual unfurling of a rosebud. The tightly packed petals slowly separate. They bend backwards. They reveal the delicate reproductive organs at their centre. The flower is opening for business. It is advertising its presence to the pollinators. The sunflower, a giant composite flower, provides another famous example of solar tracking. The young flower head will follow the sun across the sky all day, and then, at night, it will slowly reorient itself to face the east again, waiting for the dawn. It is a living clock, a floral compass, its movement perfectly synchronized with the rhythm of the day. Some plant movements are so fast that they are visible to our own naked eye. The most famous example, of course, is the Venus flytrap. The snapping shut of its trap is one of the fastest movements in the entire plant kingdom, a process that takes less than 1/10 of a second. This is an active and energy-intensive movement, powered by a sudden change in the turgor pressure of the cells in the leaf, a hydraulic mechanism that is triggered by an electrical signal. Another example is the sensitive plant, Mimosa pudica. When you touch one of its leaves, it will instantly fold up its tiny leaflets. This is a defensive response, a startling movement that may serve to dislodge a small insect herbivore, or to make the leaf look smaller and less appealing. This movement, like that of the Venus flytrap, is also controlled by a rapid electrical signal and a sudden change in water pressure in specialized structures at the base of the leaflets. But the vast majority of plant movements are too slow for us to perceive. The opening of a flower, the tracking of the sun, the climbing of a vine, the growth of a root, these are all forms of movement. They are all purposeful, adaptive, and intelligent actions. They are the plant's way of behaving. The plant is not a passive object, being acted upon by the world. It is an active agent, constantly, deliberately, and successfully manipulating the world to its own benefit. To contemplate the movement of a plant is to be forced to reconsider our own biased perception of time. Our consciousness, the consciousness of a fast-moving, warm-blooded predator, operates on a timescale of seconds and minutes. The consciousness of a plant, a slow, sessile, and photosynthetic organism, operates on a timescale of hours, days, and seasons. Theirs is a world of patient, deliberate, and graceful action. By using the tool of time-lapse photography, we are in a sense translating their language of movement into our own. We are speeding up their world so that we can perceive the rich, dynamic, and beautiful dance that is happening all around us all the time. It is a dance of life, a dance of growth, and a dance of quiet, profound, and patient intelligence. In the grand, established order of the natural world, the roles seem clear and immutable. Plants are the producers. They are the silent, passive foundation of the food chain, the autotrophs that create their own food from sunlight, air, and water. Animals are the consumers. They are the active, mobile heterotrophs that derive their energy by eating other organisms. Plants are the eaten, and animals are the eaters. It is a fundamental and seemingly unbreakable rule. But the world of life is a world of endless creativity and surprising exceptions. In a few strange and fascinating corners of the plant kingdom, this fundamental rule has been turned on its head. In the nutrient-poor soils of bogs and swamps, a group of remarkable plants has evolved a radical and audacious new strategy for survival. They have become carnivores. They have learned to trap, to kill, and to digest animals. This is the story of the carnivorous plants, a story that challenges our most basic definitions of what it means to be a plant, a story of a world where the eater, for a brief and startling moment, becomes the eaten. The evolution of carnivory in plants is a beautiful example of adaptation to a specific and challenging environment. Carnivorous plants are not found in rich, fertile meadows or forests. They are found in places like peat bogs, sandy wetlands, and acidic swamps. The soil in these environments is typically waterlogged and highly acidic, which means that the normal process of decomposition is very slow. As a result, the soil is extremely poor in the essential mineral nutrients that plants need to grow, particularly nitrogen and phosphorus. A normal plant would struggle to survive in such a place. But the carnivorous plants have evolved a brilliant, if gruesome, solution to this problem. They have learned to supplement their nutrient-poor diet by capturing and consuming a rich, alternative source of nitrogen and phosphorus, the bodies of small animals, mostly insects. It is important to understand that these plants are still, first and foremost, plants. They all have chlorophyll, and they all perform photosynthesis. They still get all of their energy from the sun. Their carnivory is not about energy, it is about nutrients. It is a form of specialized, targeted fertilization. The insect is not a meal, it is a vitamin pill. The plant kingdom has independently evolved the ability to trap animals at least 6 different times, resulting in a wonderful diversity of trapping mechanisms, each one a masterpiece of evolutionary engineering. These traps can be broadly divided into 2 categories: active traps, which move to capture their prey, and passive traps, which rely on a clever stationary design. The most famous of all carnivorous plants, the Venus flytrap, Dionea muscipula, is the master of the active trap. Its trap is not a flower, but a highly modified leaf, a bilobed, jaw-like structure with a fringe of stiff, interlocking cilia around the edge. On the inner surface of each lobe are 3 tiny, delicate trigger hairs. As we have seen, this is a smart trap. It requires 2 separate touches in quick succession to spring, a mechanism that ensures it does not waste its energy on false alarms. When an unsuspecting insect, attracted by the sweet nectar that the trap secretes, crawls across the surface and brushes against these trigger hairs. It sets in motion a remarkable chain of events. The mechanical stimulus of the touch is converted into an electrical signal, an action potential, that travels across the surface of the leaf. This electrical signal triggers a sudden and dramatic change in the turgor pressure of the cells in the midrib, the hinge of the trap. Water is rapidly pumped from the cells on the inside of the lobes to the cells on the outside. This causes the lobes to rapidly change their shape, from convex to concave, snapping shut with incredible speed in less than 1/10 of a second. The interlocking cilia on the edge of the lobes form a cage trapping the insect inside. At first, the trap does not close completely. It leaves small gaps between the cilia. This is another clever energy-saving adaptation. If the trapped prey is too small, it can escape through the gaps. The trap has decided that a small insect is not worth the significant energetic cost of digestion. But if the prey is large enough, it will continue to struggle inside the trap, repeatedly stimulating the trigger hairs. This continued stimulation is the signal for the trap to seal itself completely, forming a tight, hermetic and stomach-like pouch. The glands on the inner surface of the leaf then begin to secrete a cocktail of powerful digestive enzymes, including proteases and nucleases, which are remarkably similar to the enzymes in our own stomachs. Over the course of the next 5 to 12 days, the soft parts of the insect are slowly dissolved and absorbed by the leaf. When the digestion is complete, the trap reabsorbs the digestive fluid and slowly reopens, revealing the dry, empty exoskeleton of its victim, which is then washed away by the rain. The trap is now reset, ready for its next meal. Another type of active trap is the bladderwort, Utricularia. These are aquatic or terrestrial plants with a network of tiny, hollow underwater sacs or bladders. Each bladder is a sophisticated, high-speed suction trap. The plant actively pumps water out of the bladder, creating a partial vacuum inside. The entrance to the bladder is sealed by a small, flexible trapdoor that is hinged at the top and is held shut by the external water pressure. Near the door are several hair-like trigger bristles. When a tiny aquatic creature like a water flea or a mosquito larva swims by and touches one of these bristles, it causes the trapdoor to buckle. The seal is broken, and the external water pressure instantly forces the door to swing inwards, sucking the unsuspecting prey into the bladder along with a rush of water. The whole process takes less than a millisecond, making it one of the fastest movements in the known biological world. The door then swings shut, the plant begins to secrete digestive enzymes, and the excess water is slowly pumped out, resetting the trap. The passive traps are no less ingenious. The pitcher plants, which are found in several different and unrelated plant families, are a beautiful example of the pitfall trap. Their leaves are modified into a deep vase-like or pitcher-like structure. The rim of the pitcher, the peristome, is often brightly coloured and secretes a sweet nectar to attract insects. But this rim is also incredibly slippery, especially when wet, its surface covered in a microscopic ridged texture that prevents an insect's feet from getting a grip. The insect, lured by the promise of nectar, lands on the rim, loses its footing, and tumbles down into the pitcher. The inner walls of the pitcher are often coated with a waxy, flaky substance that makes it impossible for the insect to climb back out. It struggles for a while, and then, exhausted, it falls into the pool of liquid at the bottom of the pitcher. This liquid is a deadly cocktail. In some species, it is simply collected rainwater and the insect drowns. The plant then relies on a community of bacteria and other microorganisms living in the pitcher to decompose the prey, releasing the nutrients which the plant can then absorb. In other species, the plant itself secretes digestive enzymes into the fluid, turning the pitcher into a true external stomach. Another common type of passive trap is the flypaper, or sticky trap, used by plants like the sundews, Drosera, and the butterworts, Pinguicula. The leaves of a sundew are a beautiful and deadly sight. They are covered in dozens of long hair-like tentacles, each one tipped with a tiny glistening droplet of a thick, sticky, glue-like mucilage. An insect, mistaking these droplets for dew or nectar, lands on the leaf and is instantly ensnared, like a fly on flypaper. As the insect struggles, it only becomes more entangled. The struggling of the prey then triggers a slow, deliberate movement. The nearby tentacles, and in some species, the entire leaf itself, will slowly curl around the insect, maximizing the contact between the prey and the digestive glands on the surface of the leaf. The plant then secretes its enzymes and slowly absorbs the nutrients from its helpless victim. To contemplate the world of the carnivorous plants is to be filled with a sense of wonder at the sheer, boundless creativity of evolution. It is a world that subverts our expectations and challenges our neat and tidy categorizations. These plants are a testament to the power of natural selection to find novel and surprising solutions to the ancient problem of survival. They are a reminder that the boundary between the plant and the animal kingdom is not as clear and as absolute as we might think. In the quiet, nutrient-poor bogs of the world, the plants have learned to fight back. They have become the hunters, the predators, the silent, patient, and beautiful assassins of the green kingdom. Let us now journey from the waterlogged, acidic soils of the bog to the driest, most sun-scorched, and most inhospitable environments on our planet, the deserts. A desert is a world defined by a single, overwhelming, and life-threatening reality, the absence of water. It is a place of extreme temperatures, of relentless, desiccating sunlight, and of vast, empty spaces. For most plants, the desert is a realm of certain death. The strategies that work in a temperate forest or a lush meadow are utterly useless here. To survive in the desert, a plant must be a master of conservation, a fortress of resilience, and a paragon of patience. The story of the desert plants is a profound meditation on the power of life to adapt, to endure, and to find a way to flourish in the most extreme and unforgiving of conditions. The fundamental challenge for a desert plant is the same challenge faced by all land plants, but magnified to an almost impossible degree: how to obtain and to hold on to water while still being able to take in carbon dioxide for photosynthesis. The solutions that desert plants have evolved to solve this problem are a masterclass in biological engineering and elegant minimalist design. One of the most common and successful groups of desert survivors are the succulents. The word succulent comes from the Latin word for juice or sap, and it refers to plants that have evolved thick, fleshy, water-storing tissues in their leaves, their stems, or their roots. The cactus is the most iconic of all succulents. A cactus is in essence a living water tank. Its entire structure has been radically redesigned to minimize water loss and to maximize water storage. Let us contemplate the beautiful, architectural form of a saguaro cactus. The first thing we notice is that it has no leaves. In a cactus, the leaves have been modified through evolution into sharp, protective spines. This is a brilliant dual-purpose adaptation. The absence of broad, flat leaves dramatically reduces the surface area from which water can be lost to transpiration, and the spines provide a formidable defense against thirsty desert animals that might try to break into the cactus's precious water store. The photosynthesis in a cactus does not happen in the leaves, but in the thick, green, fleshy stem itself. The stem of the cactus is also a marvel of design. It is covered in a thick, waxy cuticle that is almost completely waterproof. The ribbed accordion-like structure of the stem allows it to expand and to contract like a bellows. After a rare desert rainstorm, the cactus's shallow, wide-spreading root system can quickly absorb a huge amount of water, and the stem will swell, its pleats expanding to accommodate the new volume. During the long, dry periods that follow, the cactus will slowly use up this stored water, and the stem will gradually shrink. The cactus is a living cistern, designed to make the most of every single precious drop of rain. But the most remarkable adaptation of the cactus, and of many other desert succulents, is a complete re-engineering of the fundamental process of photosynthesis. Most plants open their stomata, their leaf pores, during the day, when the sun is out, so they can take in carbon dioxide. But for a desert plant, this would be suicidal. Opening its pores in the heat of the desert day would lead to a catastrophic loss of water, and so the succulents have evolved a different kind of photosynthesis, a different schedule of operations. It is called crassulacean acid metabolism, or CAM photosynthesis. A cam plant keeps its stomata tightly sealed all day long, preventing any water from escaping. Then, in the cool of the night, when the air is more humid and the risk of dehydration is much lower, it opens its pores. All night long it breathes in carbon dioxide. But it cannot use this carbon dioxide for photosynthesis right away, because there is no sunlight. So it fixes the carbon dioxide into a temporary chemical storage form, a 4-carbon organic acid called malic acid. It stores this acid in the vacuoles of its cells. The plant literally becomes more acidic during the night. Then, When the sun comes up the next day, the plant closes its stomata again, sealing itself off from the dry outside air. Now, inside its own, closed, humidified tissues, it can perform the second part of the process. It takes the malic acid out of storage and releases the carbon dioxide. This carbon dioxide is then used in the normal Calvin cycle, powered by the energy of the sunlight that is now streaming down, to produce sugars. The cam plant has separated the two parts of photosynthesis in time. It takes in the carbon dioxide at night, and it processes it during the day. It is a strategy that is far less efficient than normal photosynthesis, and it leads to very slow growth. But in the extreme environment of the desert, it is a strategy that allows the plant to survive. Not all desert plants are succulents. Another common strategy is to become a drought deciduous shrub. Plants like the creosote bush or the ocotillo have small waxy leaves during the brief wet seasons. But when the long dry season begins, they simply drop all of their leaves and enter a state of deep dormancy, appearing as a collection of dry, dead-looking sticks. They are not dead. They are just sleeping, waiting. They have shut down their metabolism to an absolute minimum, conserving the precious water in their stems and their roots. Then, when the rains finally return, they can burst back into life with incredible speed, growing a new set of leaves in just a few days. And then there are the most ephemeral and patient of all desert plants, the desert annuals. These plants are the drought evaders. They do not try to endure the dry season at all. They survive the long, hot and waterless months not as a living plant, but as a dormant, patient seed, buried in the dry soil. These seeds are masters of waiting. They can lie dormant for years, or even decades, waiting for the one specific signal that will tell them that conditions are right. They are waiting for a rain that is heavy enough to wash the germination-inhibiting chemicals from their seed coats and to provide enough moisture for them to complete their entire, accelerated life cycle. When that perfect rain finally comes, the desert floor can erupt, almost overnight, into a breathtaking, super bloom of colour. The seeds all germinate at once. The plants grow with incredible speed. They flower, they are pollinated, and they produce a new generation of seeds, all in the space of just a few, short weeks before the soil dries out again. They have compressed their entire life into a brief, beautiful and frantic window of opportunity. They have gambled everything on a single perfect moment. To contemplate the life of a desert plant is to be filled with a profound sense of respect for the power of resilience. It is to see that life is not just about abundance and growth and speed. It is also about patience, about conservation, and about the quiet, stubborn and beautiful art of holding on. The desert survivors are the stoics of the plant kingdom. They have stripped themselves down to the bare essentials. They have learned to live with less. They have learned to wait. And in their stark minimalist and architectural beauty, they offer us a powerful lesson in the quiet strength and the profound, unyielding tenacity of life.